Hi, everybody. I'm presenting today work on validating diffusion MRI with polarization-sensitive OCT. This is work by my graduate student, Georgia Grisot, who could not be here today because she has to give another talk at ISMRM on her other diffusion validation project. And as an overview, we're following these two complementary approaches to validating diffusion MRI at the moment. On the one hand, the work that Georgia is presenting at ISMRM, we are collecting high-resolution ex vivo diffusion MRI data on macaque brains that have received tracer injections, and where, based on the histological processing of the tracing, we know all the parts of the brain that are connected to the injection site, and so this is appropriate for validating tractography in the diffusion data from the injection site. But it does not allow us to validate fiber orientation distributions, for example, because at every voxel in the image, we have the axons that are connected to the injection site, but not all the other axons that are crossing. On the other hand, in the work that I'm presenting today, we have direct measurements of axonal bundle orientations at microscopic resolution from optical imaging, and this does allow us to reconstruct all the different fiber populations at each voxel and compare to diffusion orientations. This at the moment is not feasible, however, on the whole brain, and so that's why we cannot use this to validate long-range tractography. Polarization-sensitive OCT takes advantage of birefringence, which is an optical property of anisotropic tissues, and it is the difference in refractive indices parallel versus perpendicular to the axis of anisotropy. In the brain, it occurs due to the regular pattern in which arrays of lipids and proteins are arranged in the myelin, and so here the optical axis is that of the myelin sheath, and hence also the axons. In PS-OCT, we use polarization of light to probe this birefringence phenomenon. And in addition to the conventional reflectivity contrast that we get from conventional OCT, we also get images of the optic axis, which is an estimate of the fiber orientation in the plane orthogonal to the incident light. And we can collect data with multiple incident angles to reconstruct 3D orientation. And we also get what's called the retardance map. And this is essentially a measurement of how much myelin is there and can be used to register to MRI scans. Unlike myelin stains, PS-OCT does not require any kind of image processing of histological images to extract the fiber orientations, because the fiber orientations come to us as a direct measurement of the scan. And unlike both myelin stains and polarized light imaging, PS-OCT does not suffer from histological type nonlinear distortions. So the slices come out perfectly aligned to each other because we image each slice, we image the superficial layer of the tissue based on the light that's backscattered from the tissue, and after we image it, then we cut it and move on to the next slice. As we cannot image the whole brain at the moment, we have to go after specific regions in the white matter, and we choose them motivated by our monkey tracer validation studies. In particular, we locate brain areas where in vivo diffusion tractography in humans tends to take paths that are not expected based on the monkey tracing data, and we have no way of knowing if these are true interspecies differences or errors in the diffusion data. The example that I'm going to show is from an area of white matter where we expect fibers from the prefrontal cortex to split into two bundles, one going through the corpus callosum and one going through the internal capsule. But sometimes, as in the example that I'm showing here, in human in vivo tractography data, we see that the internal capsule bundle is missing, and instead the tractography takes a different path. We locate that particular area where that split is supposed to happen in a postmortem human brain hemisphere. And we do that based on a whole brain, or whole hemi in this case, 750 micron resolution scan, using the tractography data in that whole hemi scan to find that block and then cut it out of the actual brain. Then we take that block and we image it at much higher resolution, 250 microns isotropic, in a Bruker 9.4 Tesla small bore scanner with B values all the way up to 40,000, which is roughly equivalent to 10,000 in vivo. We collect the data on a Cartesian grid acquisition scheme in QSpace, and then we use that data and the non-uniform Fourier transform formulation to generate data on Q shells for the types of analysis that require that. And I'm going to show comparisons of 5 different analysis approaches. One is diffusion spectrum imaging, which requires grid data. One is generalized Q-space imaging, which is a model-free ODF-based method that works on either grid or shell data. And another ODF-based method, Q-ball imaging, using the constant solid angle derivation, which requires shell data. And finally, 2 model-based approaches, the ball and stick and the tensor model, which in principle could be fit to either type of data, but shell data fit them better. So once we have the diffusion data, we find the most complex and most interesting area of the block, and we perform a PS-OCT scan on it. I'm showing here a scan that was collected with 10 micron in plane and 75 micron through plane resolution, and you can appreciate that the slices come out perfectly aligned to each other, so there's no histological nonlinear registration to do here. What I'm showing here is on the upper left, the PS-OCT measurements of the orientation of the optic axis at 10 micron resolution, and then the others are the diffusion orientations. Each column is a different diffusion analysis approach, and each row is a different level of spatial resolution, going from the native resolution of 250 microns at the top, and then downsampling to 0.5 and 1 and 2 millimeters. What I'm showing here is the results of the DSI and GQI reconstructions on the full grid data, and we see that we're doing a pretty good job recovering the colors that we see in the ground truth PS-OCT data. And things remain reasonable up down to a resolution of 1 millimeter, more or less, and then somewhere between 1 and 2 millimeters is where things really break down. And this suggests that 1 millimeter resolution could be a reasonable target and a potentially feasible in vivo target to go after in the next few years. Now, looking at what we get when we fit the ball and stick and tensor models in the same data, we're not doing quite as well. So I want to draw your attention to this area in the upper right, where both in the ground truth PS-OCT data and in the DSI and GQI reconstructions, we see a mix of red and yellow orientations. You'll see, if you check the color wheel at the bottom left, that these are actually 2 orientations that are pretty close to each other. With the ball and stick and DTI, we're not able to resolve them, so we see a sea of red in that area. And similarly, in this area in the left, where in the PS-OCT as well as the DSI data, we see a mix of blues and greens, the ball and stick and DTI show mostly blue. So one could say that we're disadvantaging those models because we feed grid data to them. So let's see what would happen if we feed the single shell data that are supposed to be a better fit to these models. And I'm going to flip back and forth. You see that as I go between the grid and the shell data, now we are starting to see more differentiation in those areas. We see some of the orange emerging in the red area, and we also see some differentiation in the blue area. However, there's all the cyan stuff that's not really there in the ground truth data. So we're doing a little bit better because we're using a Q-space acquisition scheme that's better, a better fit for these models. But actually, as I'm going to show in a second, we can do a better job on this single shell data if we use the ODF-based model-free approaches. And so this is the GQI and the QBI on the single shell data. And you'll notice that these look much more similar to the DSI and to the ground truth OCT data than the ball and stick and DTI. And I'm going to flip between the single shell and the grid just to show that actually GQI does a better job when it uses the full grid data. Now, what if we had multiple shells? Do we gain anything? So this is the 3-shell data, and we do see some cleaning up, particularly for the ODF-based methods. So the GQI and the QBI get closer to the DSI and to the PS-OCT data when we use multiple shells. There is a little bit of an improvement for the ball and stick. We see more of that yellow inside the red. But the same general trends are seen here, that things are reasonably good up until a 1 millimeter spatial resolution, and then things break down. And next, I'm going to show you another sample that is more complicated than this. So here, even though we have that area of the splitting between the yellow and the red orientations, in general, things are clustered. We see a green area, a red area, and a blue area. And I'm going to show you another sample This one is close to the edge of the caudate, but still in white matter, where we really have these fiber populations that are really intertwined with each other and crossing. So what you see at the top left is the orientation axis image from the PS-OCT, and right under that is the intensity image from the OCT. So you see that really this is a very complex area of white matter. And interestingly enough, at the native resolution of 250 microns where we're collecting the diffusion data, both the DSI and the GQI reconstructions of the full grid data are doing a pretty decent job capturing the colors that we see in the PS-OCT. The other methods don't do quite as well in this very complex sample. So QBI on a single shell acquisition does okay. Ball and stick didn't really do very well on this sample in any acquisition scheme. Going forward, we would like to further investigate the grid acquisition by potentially undersampling it and using compressed sensing approaches to speed up the acquisition and potentially go to even higher resolution. But these images are encouraging us that if we could collect at 250 micron resolution ex vivo images of the whole brain, we could get very close to having a ground truth atlas of the wiring of the brain. And on the PS-OCT side, there are efforts underway to build a new rig that can collect larger field of view, even going up to the whole brain. And in the more immediate term, collect data with multiple incidence angles to validate 3D rather than just in-plane orientation. And I would like to end by thanking Georgia and Robert, who did the analyses that I showed, Huave, who built the PS-OCT rig that we used to collect this data, my other co-authors, as well as NIBIB for funding this research. Thank you.